শুনা পালা ন? Okay, then let's start the class. Lecture কিমান হ'ব? Class number? লিখিছা নেকি কোনোবাই? Class number কিমান? 3rd নেকি? কোনো লিখা নাই? Lecture number? 4. Sure, no? Thank you. অকমান লিখি থবাচোন সাইডত, মই কেতিয়াবা পাহৰি যাওঁ, বহুতবোৰ ক্লাছ চলি থাকে তো। Okay, so in the last class what we have discussed was we have started with the first kingdom, that is Monera, and we have started with the discussions about the best example, that is bacteria, right? Different types of Monera, and we have started with something called as eubacteria, right? Bacteria both types, kintu kisuman group of we use the word eubacteria or they are called as true bacteria, right? What we have discussed, we have talked about the cell envelope in the last class and the structure of flagella and classification of bacteria to some extent, right? Structure of flagella, I think we remember that it has 3 parts and all those rings and all those, right? So that was the portion that we have discussed. Today we will be starting with something called as life process of bacteria, right? Eta bacteria jibonot ki reactions hoy ba ki life process chole, right? So today's topic would be and under this topic, the things that we are going to read is right? So life process of a চাৰিটা বস্তু we should read, right? Or we should have an idea. Yes, was there anyone who was asking me something? কোনোবাই সুধিছিলা নেকি online? Okay. So we'll start with the first one. When we talk about bacterial growth, we understand that a bacteria could be found almost each and every region. We have already discussed something called as ubiquitous in our previous classes. Do you remember that word called as ubiquitous? Right? Ubiquitous ki hoy je bacteria jedi sobote poa jay no, it is universally present, and so we are using the word called as ubiquitous. It is present inside the body of an organism. It is also found outside the body of an organism, right? So talking about bacteria, it could be found in each and every place. It can grow in each and every place, inside or outside, in very hot areas or in very cold areas, etc., etc. Obviously next is the two words natural and artificial. Naturally obviously they are found, and artificially also we can lead to the growth of bacteria. Eta bacteria ami artificially grow koribo paru, right? And when we are talking about growing bacteria or developing bacteria in artificial medium, we need to talk about something called as culture medium, right? Because if you want to grow a bacteria, first of all, what do you understand by growing of bacteria? We have already discussed in the living world class, right? If I say a bacteria is growing, what we understand that the process of cell division is occurring in the bacteria. One bacteria, it becomes 2, 2 পৰা কেটা হয় 4টা 4টাৰ পৰা 8টা হয় এই যে increase in number, this is nothing but the growth of bacteria. It is not like this that the bacteria will keep on growing in size and ultimately it will become a huge structure, তেনেকুৱা নহয়, right? So if you are talking about growth of a bacteria in artificial medium or artificially or in the laboratory, we talk about culture medium. And talking about culture medium, different types of culture medium could be used for the growth of bacteria. Culture mediums, for example, it could be non-synthetic culture medium. When we use the word non-synthetic, we understand we are talking about the natural culture medium. For example, natural culture medium, jikunu eta example, milk, juice, right? Milk auto bacteria hobo pare, juice auto bacteria hobo pare, enukwa jodi natural products bilak ami use koru no, and we use it for the growth of bacteria, those are categorized under non-synthetic culture medium. Similarly, we have synthetic culture medium also, something that we prepare in the labs and then we put in a petri dish and then we help or we allow the bacteria to grow. And together with that, natural but non-synthetic culture medium, synthetic culture medium, and also in certain types of embryonic tissues also, right? As we have already discussed, bacteria could be found in each and every place. Jote tote hobo pare, kisuman organs auto hobo pare, organic mediums to hobo pare, embryonic tissues auto hobo pare. So ultimately the one line that we remember is it could be found almost everywhere. It could be grown artificially, it is found naturally also, right? So in the PDF there are descriptions about the different types of culture medium. Examples bilak diya ase, not very important from examination point of view, karon ketiao etu question has never been asked that give an example of a artificial culture medium, especially in this chapter, right? Or give me an example of a culture medium where you can grow bacteria. Enukwa dhoroner questions aha nai. This is just a point that you have to go through. See the PDF, just read it once and that's all. You don't have to buy hard. Next, talking about the growth, you always, each and every organism have something called as optimum requirement, right? Bhalke grow koribo lagile optimum but best, no, kiba eta features lagibo. So if you are talking about temperature, there should be a best temperature for the bacterial growth. If you are talking about acidity or if you are talking about pH also, there should be a best pH where we see the growth of bacteria is maximum. So hegita point to likha ase, it's almost 35 degrees Celsius where the bacteria grows best. If you are talking about pH, we have seen that it's 7, pH number 7 on basic grow kore. So there are certain informations. What you remember is just the temperature. Temperature point 2, already it's written there, so we will not write it down. Next is par kataman point likhi dim. Next is once we are discussing about the growth of bacteria, if you study the growth of bacteria, the study or the result that we see could be depicted in the form of a graph, right? Suppose we are talking about a bacterial growth maybe in a laboratory. It's in a petri dish that the number of bacteria keeps on growing. Prothomote ki asile? Eta asile, and after that the number kept on increasing. If you are going to study this data and put it in somewhere or in the form of a graph, we are going to obtain something called as a curve, right? Whenever we talk about a graph korar ami curve dekha pao. So in case of bacteria also, there is something called as a bacterial growth curve, right? So when we talk about the growth curve, what is the growth curve? If I use a word called the sigmoid curve, is the point understood? Right? If I say bacterial growth could be represented in the form of a sigmoid curve, is the line understood? Sigmoid curve ki hoy? Organism and population, we have already completed the chapter. We have talked about two types of curves, logistic and the J shape, right? Exponential or logistic. One was S shaped and the other was J shaped. That S shape is called as ki sigmoid, right? And when we talk about sigmoid or the S shape, that kisuman phase thake, no? Ki asile leg phase, log phase, exponential phase, stationary phase, right? So when we are talking about bacterial growth is represented through which curve? It is represented through a sigmoid curve or a S-shaped curve. So these are the only 3 or 4 points that you have to remember from this portion, right? So ki likhiba, details already given in the PDF, so we don't write it down. Dutaman point likhim khali. Eto topic, huh? continuation. Sorry. Living cultural medium word to বুজি পাইছো ন right so cultural medium হল আমি living cultural medium বুলিও ক'ব পাৰো যে living tissues or ওপৰত grow কৰে বা use it as a medium for growth that is called as living cultural medium. Okay, so this is a part that we have already discussed, so I will not write anything in details. We understand if you are talking about lag phase or log phase, question 2 is highest growth of bacteria occurs in which phase? It is always in the log phase. If you are talking or if you are using the word called as lag comes from the word called as lagging behind, right, where the growth of the bacteria is less or fewer in number, because if you are talking about the first bacteria that is growing at the initial period, the number of bacteria that would be formed by cell division would always be less, because if you are starting from one or pra keta hobo, it's just 2 that we will get, right, and if you are talking about 2, the next stage we are going to get 4, but at a certain time, suppose we are talking about 16, right, 16 to ki hobo directly double hoy pelai 32 hoy jabo, 32 will again directly double and become 64. So we see that all of a sudden the number has already increased, right, and that sharp increase is what is called as exponential growth or the highest growth. Highest growth is seen in case of log phase. Lagging behind, the growth is very less. Talking about the stationary phase, I think we understand the meaning. It's the phase where the growth becomes almost constant. And what is the reason behind becoming constant? Maybe because of the requirements or lack of requirements. The food could be a problem, the space could be a problem. Ekhon petri dishot grow kori asile eta duta jetia asile tetalke problem nai, no, if it is 1 and 2 it's not a problem because it has enough food material or space to live. But if the number keeps on increasing in a definite habitat or in a definite area or in a particular petri dish, what happens? If it keeps on increasing, at a time there will be a competition for food, space, etc., and then obviously, as we have already discussed, competition hole survival of the fittest hobo aru kisuman mori jabo, and ultimately once the entire food is used up, it will lead to the death of the bacteria. So this is all about growth, nothing very important, kintu etu point monot rakhiba ha, etu pra question ahise this part. So this was about the first life process. Next we go to the next one, that is respiration. Heading koriba respiration. Again, from respiration part, whenever we use the word respiration, we have to remember the types of respiration. Talking about types of respiration, aerobic and anaerobic. Similarly, the same explanation for bacteria also, aerobic respiration and anaerobic respiration. Aerobic respiration means the respiration that takes place in presence of oxygen is called as aerobic. In absence of oxygen, that's called as anaerobic. But together with aerobic and anaerobic, we'll be using two other terms, that is obligate and facultative. Right. So, respiration divided into two categories, that is aerobic and anaerobic. Aerobic divided into two and anaerobic again divided into two categories. What is the question that is asked? From this part, the question that is asked is about the examples. Right. So, question could be give an example or identify the example of facultative aerobic bacteria, or obligate aerobic bacteria, or obligate anaerobic bacteria. So, obviously, I will write the chart, okoman chart ekon ase, that examples important boli likhi thoba, right. Students, please see if you can follow the spellings. Online students, spellings follow koribo parisan nai, because examples are obviously important. Are you done? Likha hol ke? Okay, talking about respiration, aerobic and anaerobic are the two terms that we understand, right, in presence of oxygen, in absence of oxygen. But we will be using two other words, one is obligate and the other is facultative. What is obligate? Obligate means strict, right, and what is facultative? Someone that can switch over, that is called as facultative. Ohomiyat amar eta word ase no, subidhabadi, right. So, what happens is, you can adjust yourself in any conditions. You can stay here, you can stay in the other room. Suppose we are taking the examples of two rooms, right? Suppose this is a room, another there is a room. In this classroom, AC to soli asile, all of a sudden the AC stopped working. So what you will do is either you stay in the classroom in spite of the fact that the AC is not working, or you will move to another classroom where the AC is working. So there are two conditions. Either you will adjust and stay there until your death, or what you will do is you will keep on adjusting. If you keep on adjusting, that is what is called as facultative. Ohomiyat ki ko subidha badi. Subidha pale iyat thakibo nohole tat thakibo. Right? But if you are using the word obligate, that is a strict one. It will never change its place and never change its requirements also. Right? So obligate aerobes. So what we understand that these are the group of bacteria who can survive or respire only in the presence of oxygen. If there is no oxygen, then what will happen to these organisms? They will die. Clear? So that is the reason why it is called as strict. But if you are talking about facultative, the first point to notice here is we are not writing facultative aerobes. Instead, we are writing a word that is called as facultative anaerobes. Whenever we are doing a classification to jimanbur types sake, no, it is always under a particular heading. If suppose we are talking about stems, types of stems, aerial stems, weak stems. So we always use the word stems. If you are talking about roots, tape roots, modified roots, right? But here if you notice, we are talking about aerobic group, but instead we are writing here as aerobic and here as anaerobic. So this is something that you need to remember. You have to remember what is the difference between facultative aerobes and facultative anaerobes, right? No doubt this is a classification about aerobic bacteria. Kintu eta classok ami ki boli koisu? Facultative aerobe boli koisu na facultative anaerobe boli koisu? Facultative anaerobe boli koisu. So I think this part is understood. Coming to this part, facultative anaerobes ki hoy? These are those group of bacteria. Originally they were aerobic, but if there is a lack of oxygen, they can survive without oxygen also. Right? So they can survive in an area with oxygen, they can survive in an area without oxygen also. The question that is asked is, facultative anaerobes will originally ki asile? Are they a group of anaerobic bacteria or they are a group of aerobic bacteria? Originally they were aerobic, they were surviving in an area where oxygen was present. Due to certain conditions, it might happen that in that area the oxygen condition might get decreased or it might deplete, right? So in that case, they can switch over to anaerobic, means they have a tendency to live or survive or adjust more easily, right? That is what is called as facultative. Question kya hai? Identify the example of a facultative anaerobe. Identify the example of a obligate aerobe, right? Next, again from this anaerobic group ki hoy, the group of bacteria that can survive without the presence of oxygen is called as anaerobic. Obligate anaerobes. What will be the sentence? Ki buji paisu? This is a group of organisms that will survive purely in absence of oxygen. If you supply oxygen to these group of organisms, then what will happen? They will die because they are strictly ki? Anaerobic, right? But if you are talking about facultative aerobes, originally ki hoy? Anaerobic, but they can switch over to what? Aerobic. Similarly, in this condition, facultative anaerobes, originally ki asile? Aerobic, but they can switch over to which condition? Anaerobic. Is I mean terms understood. Facultative or obligate. Words kita buji paisu? Kiba doubts ase niki online? Okay. No. Clostridium botulinum is a genus and a species, right? I think it's over. So side note likhi thoi diba, examples important. Right. Next is nutrition in bacteria. Talking about respiration, we come across the two words aerobic and anaerobic. Talking about nutrition also, we come across two words, kiki, autotrophs or heterotrophs. So in case of bacteria also, there would be certain types of autotrophic bacteria and certain types of heterotrophic. If we are using the word autotrophic, then we understand that we are talking about what? The organisms that will produce their own food. As far as producing their own food is concerned, producing food, we often refer to a process called as photosynthesis in plants, right? So if some organism has to prepare its own food, then obviously there would be certain types of requirements for the process. So photosynthesis of kisuman requirement asile tetiahe food banabo pare. Similarly, in case of bacteria also, if it has to prepare its own food, it will have its own requirement. If the main requirement is light, we will categorize under the heading called as photosynthetic bacteria. If it gets all the energy from chemical substances or because of chemical reactions, we will be categorizing under the topic called as chemosynthetic bacteria. So obviously we will have certain terms. Again, what is important is examples are important. Right. So question ahibo pare, identify the example of a photosynthetic bacteria, identify the example of a chemosynthetic. Again, photosynthetic and chemosynthetic have its own categories. Right. Photosynthetic or duta category ase, chemosynthetic or duta categories ase. We will be talking about. So prothomote likhi loa explanation pison. Autotrophs, heterotrophs, we understand those that can prepare their own food, those that they cannot prepare their own food. If an organism cannot prepare their own food, then from where will they get the food? Buji paisa ne bostu tu? Nijor khwa bostu nije banabo pare, it's very good. If you can't prepare your own food, then obviously you have to depend on someone else. That someone else, karpora food to loise, depending on that we have certain categories. I think you have heard certain words called as parasites, saprophytes, etc., etc. What is the difference between a parasite and a saprophyte? If the organism takes the food from a living host, then it is called as a parasite. Or if dead host or praloy, then it is called as a saprophyte. So what we need to remember, heterotrophs or kisuman types ase. So heterotrophs divided into three categories, either it will be a parasite or it will be a saprophyte or it will stay in a symbiotic association. Parasite we understand, saprophytic we understand, symbiotic association mane? Mutual collaboration, right? Both of the organisms will get benefited. We have already talked about something called as mutualism. Organism and population or mutualism is something that we have discussed, right? So examples. So once this is over, we will be talking about the autotrophs. Ekhini hol niki? Autotrophic chart to okman dangor hobo de. It will be comparatively long. Either you can write it in the form of a chart or you can write it in the point wise also. Over hoise? প্ৰথমতে লিখি লোৱা যোনটো ৱৰ্ড আণ্ডাৰলাইন কৰিছোঁ সেইটো আণ্ডাৰলাইন কৰি ল'বা over so from examination point of view or question point of view it is the examples that you need to remember right concept to aru iman nalage kintu tothapi jedi kiba concept wise question ahe so obviously we have to understand each and every term right kintu questions ki aise if you go to the MCQ books you will see that only the question has been asked about that examples or maybe there could be a one example so if you are talking about the difference between a lithotrope and a organotrope what is the basic difference you have to remember organic substance and inorganic substance right is the chart over okay talking about the explanation talking about nutrition, autotrophs, heterotrophs, we understand autotrophs ki hoy, those that can prepare their own food are called as autotrophs, right? As I said before, if it is an autotroph, it will have its own requirement. Whenever we talk about autotroph, generally photosynthesis is the word that we remember, right? So similarly, in case of bacteria also, there will be certain types of photosynthetic bacteria, there will be certain types of chemosynthetic bacteria. What is the basic difference? That if they prepare their food in the presence of light, light being an important factor, obviously whenever we talk about food, sobotse basi important raw material ki hoy. If you are talking in case of plants, right? Plants undergoes photosynthesis and it has its requirements, pasta type of requirement, that's it. Then only they can prepare their own food. Just say, give me some examples of the requirements. It would be carbon dioxide, it will be water, it will be light, right, sunlight, right, then it will be that structure where the photosynthesis process occurs, that we are talking about the chloroplast, and obviously the last one is obviously the pigments. These are the 5 requirements for a plant to undergo photosynthesis. Tar birat carbon to lagiboi aru different things. Okay, next is, so they will, if they prepare their food in presence of light, those are called as photosynthetic. Group of bacteria that produce their food in presence of certain types of chemicals or by obtaining the energy from a chemical reaction, then it is called as chemosynthetic. First, the difference. Duita difference buji paiso? Light lagibo le light na lage. Jedi light lage, photosynthetic, purely dependent on light. Light thakilhe food bonam, photosynthetic. If there is no light, they will take all the requirements from some certain types of chemicals, it is called as chemosynthetic. We understood. Next, talking about photosynthetic again divided into 2 categories, and chemosynthetic has again been divided into 2 categories. The first differentiation was on the basis of presence of light or absence of light, 2 category. Next category is on the basis of what? On the basis of a source of electrons and protons. Right? Now you have to understand what is the role of electron and proton in the process of preparing food. You have done the chapter called as photosynthesis previously, obviously we have not done in the class. If you remember, in case of photosynthesis in plants also, for the process to undergo, but for the process of preparation of food, electrons and protons play a major role. Could you identify what was the source of electrons and protons in case of photosynthesis in plants? The first point is, does electron and proton play a role in photosynthesis? The first point, obviously yes. Second means what? Electron proton lagibo, tetiahe photosynthesis hobo, no? The plant will have to have a source of electron and proton, then only the photosynthesis will be conducted. Second, what is the source of electrons and protons and why is it necessary? Exactly. So in case of plants, ki hoy? Electron and proton or source ki, it is the water molecule. Heard something called as photolysis of water. Right, photosynthesis, you have talked about photolysis of water. We know light is important. What happens to the light once the, what happens to the water once the light falls? Water to lagibo, photosynthesis koribo lagile. So what happens to that water in presence of light? It splits. If a water molecule splits, what are the products that you get? Photosynthesis hobo lagile, photolysis of light hobo lagibo, right? If photosynthesis has to take place, then photolysis of light will also have to take place. If photolysis of water takes place, then the water molecule will break down in presence of light into what? Eta molecule bhangi gole kisman components form kore, no? Right? If a molecule gets broken down, then obviously we see certain types of components. If you are talking about NaCl, if it breaks down, Na part and the Cl part. Similarly, if water molecule breaks down, what are the parts that you obtain? First point, what is the role of water? Why is water so essential in photosynthesis? Correct. We know if you are talking about requirements, carbon is a requirement. What is the role of carbon? Ki kore? Plante carbon loy. Very good. Kiyo karone loy? Food prepare koribo lage. Okay, very good. What do they do there? Or what is the role of carbon? Carbon gets converted to what? Food itself, right? CO2 hisabe loise, kihole convert hoise, C6H12O6, right? So it is the main food that is prepared from that carbon itself, right? Together with carbon, photosynthesis reaction to ki asile, carbon dioxide plus water will give you the food. What happens to carbon? Converts itself to food. What happens to water molecule? It breaks down. Breaks down into what? If a water molecule breaks down, there are three things that are produced: electrons, protons and oxygen. Obviously H2O ase no, anything that breaks down electron proton to produce koriboi lagibo, right? So if the water molecule gets broken down, also electrons, protons and oxygen. Again, one point from the photosynthesis part, no, if you are talking about breakdown, we can write it like this: eta water molecule bhangile ki produce hoy? Electron, proton. If you don't consider, what is the main thing that it is producing? Oxygen, right? We know photosynthesis process or ki hoy, oxygen gets evolved, right? So if oxygen has to get evolved, someone should produce the oxygen. So oxygen to copra paiso, obviously from water, right? So the next point from that chapter, to obtain one molecule of oxygen, how many molecules of water should get broken down? Karon if one molecule of water breaks down, one molecule of oxygen is not obtained. Karon yate ki ase? O he ase. O e ki produce koribo? Nascent oxygen he produce koribo. Kintu if you are talking about oxygen, it's O2, right? So that is another point that you need to remember. To obtain one molecule of oxygen, two molecules of water should break down. This is a part that we will be discussing in details in photosynthesis. As of now, coming to this point, we know water is a requirement for food synthesis. When water breaks down, these are the things that get produced. Oxygen is a gas that gets evolved or cell to pra ulai jai, right? What remains is electrons and protons. Now tell me the role of electrons and protons. We know we have a cell. Inside the cell, water is present. Water ki korile in presence of light, it got broken down, ki form korile, electrons, protons, oxygen. Oxygen is a gas, goes out of the cell. What remains in the cell? Electrons and protons. If this electron and proton is left like this, they will join and again form water, right? So it's very essential that this two needs to get separated from the cell. If it has to get separated, then they have to be used up also. So you have to remember the electron ki kamot use korisile, proton ki kamot use korisile during the process of photosynthesis. That will explain what is the necessity of a source, right? I have written a word called as source, no? Each and every organism who prepares food will obviously have to have a source of electron and proton. The source might be different. It is not necessary that water will always be the source. For certain organisms, the source of electron and proton could be something else also. For certain type, it would be water. Talking about plants as the most common example, tahator source ki, electron and proton or source is always water, right? Coming to this point, oxygen, we understood what is the role of electron and proton. Electron or ki hoy, proton or ki hoy? Electron transport system or ETS. ETS, so obviously ETS, but ETS is something that you have read in the chapter photosynthesis, in the chapter respiration. Respiration, because whenever we talk about ETS is there in both the process, right? But detailed description obviously in the respiration we have talked about, no? At the diagram eneke asile, right? Eneke move korisile, no? ETS in respiration. But in case of photosynthesis also, we are talking about electron transport system itself, kintu it is slightly different from respiration and photosynthesis, right? So the point is, in case of photosynthesis, where does the electron move or what is the function of the electron? Ki form kore. It assists in which reaction of photosynthesis? Photosynthesis divided into two reactions, that is light reaction and dark reaction, right? This is something that we know. What happens in the light reaction? What happens in the dark reaction? এটাত মানে ফার্স্ট তুমি লাইট আৰু ডাৰ্কৰ কথা কোৱা লাইট ৰিঅ্যাকশন জেড স্কিন টু লাইট ৰিঅ্যাকশন আৰু সাইক্লিক টু ডাৰ্ক ৰিঅ্যাকশন জেড স্কিন টু লাইট ৰিঅ্যাকশন সাইক্লিক টু ডাৰ্ক ৰিঅ্যাকশন এট লিস্ট কনসেপ্টটো ক্লিয়ার বাকি রেস্ট এটোটো শুনিছা ন photosynthesis divided into light reaction and dark reaction, right? What happens in the light reaction, what happens in the dark reaction, and in which reaction the food is prepared? Food in the form of glucose is prepared during the light reaction, the dark reaction. For it is always in the dark reaction that the food is prepared. Food puli kwa loge loge, it is that glucose molecule or starch molecule, whatever you see, right? What you need to remember is, in order to prepare food, we need to have certain types of raw materials also. We should do something to get food. For example, in our home, if you want to eat rice, you have to bring that rice, you have to cook it, then only you will eat rice. Without cooking, you cannot have the rice. Similarly, in case of plants also, if the cell has to have glucose or starch or food, they have to have some other process before. Kiba eta koribo lagibo, tetiahe food to prepare hobo, right? And what is that thing we are talking about? We are talking about the things that occur in the light reaction, right? Light reaction not ki hoy? The raw materials required for food preparation is prepared. What is the raw material for the food? The raw material for the food is ATP and NADPH2. What is NADPH2? The reducing cofactor. Who has to get reduced? Ekini information asalte photosynthesis chapter 2 hoy. Because photosynthesis is an example of, you see reduction reaction also, you see oxidation reaction also. So you have to be clear who gets oxidized and who gets reduced. If something gets reduced, then someone has to help it, and that someone is called as a reducing cofactor. Reducing cofactor kun hoy NADPH2 in case of photosynthesis, NADH2 in case of respiration. Right? Duta word poriba, tumalage tinta word poriba, reducing cofactor, NADPH2, NADH2 and FADH2. These are the three words that you have already read. What you need to remember, NADPH2, the reducing cofactor required for photosynthesis, NADH2 and FADH2, remember that Krebs cycle, right? So these are the reducing cofactor in case of respiration, right? How will you remember NADH2, NADPH2? P eta ase no? So P is photosynthesis, right? So that is also a question that is asked. The reducing cofactor that is required during the process of photosynthesis, NADPH2, NADH2, it is NADPH2, okay. Next is, we have already said that during the process of light reaction, ATP and NADPH2 needs to be formed. Those are the products of light reaction. ATP, obviously the source of energy. NADPH2 by NADPH, kune form kore? Who forms NADPH? Light reaction or producer, we understand, right? The product of light reaction, ATP and NADPH2. Who forms NADPH2 or who forms NADP? NADPH or NADPH2 is the same thing. Either you get more reduced and another hydrogen gets attached. The main molecule to ki NADPH, right? So, jodi aru eta hydrogen log lagi jay, tetale NADPH2 hoy jabo. Okay. Now, the question is, who forms that NADPH? All of a sudden, it cannot be formed in a cell. Kunubata thakibo lagibo. There should be a raw material that gets converted to NADPH, right? It is always NAD that forms NADPH, right? So there is a molecule called as NAD. Who forms NAD? We'll talk about in the reactions in photosynthesis, not now, right? So as of now, what you remember, light reaction, dark reaction, duta ase. Light reaction or product ki ATP and NADPH or NADPH2. For the formation of NADPH2, NADPH will have to take another hydrogen. Hydrogen is nothing but a proton. If a molecule has to accept a proton, if it is in a neutral state, will it accept a proton? No. It has to remain in an excited form, right? So what happens? It takes in electrons. Electrons jetia loy, he negative hoy thake takile tak positive lage. So ultimately NADPH2 gets formed. What is the process? Details in the photosynthesis class. As of now, this electron goes to what? It goes to NAD, right? So ultimately, it helps in the production of a raw material during light reaction, right? So out of the three things, oxygen buji palu, E has a role in the formation of NADPH2. Coming back to protons now. Proton or a role thakibo lagibo. You cannot leave this proton in the cell. If it's left in the cell, then what happens? The concentration will keep on increasing, right? Light reaction or product duta, ATP or NADPH2. NADPH2 produce koribole generally, it is both of them that they will help, but maximum kune help kore electrons bilake. All electrons to jabo nware, no, because only electrons, if some molecule accept it, then it will become negative. It has to take the positive factor also. But basically, the credit we give, that we say that it helps in the formation of NADPH2. Coming to this, remember something called as a proton gradient. Proton gradient, what to hold this? Have you heard something called as chemiosmotic hypothesis? Photosynthesis chapter NCERT bookot last diagram eta ase chemiosmotic hypothesis. What does it talk about? Chemiosmotic hypothesis bola bostu tu ki bujai? It talks about ATP formation during photosynthesis, right? And ATP formation jodi hoy kot ATP form kore? ATP forms in photosynthesis, ATP also forms in respiration. If ATP has to be formed, then there should be certain requirements again. If food has to be formed, there should be certain requirements. If ATP has to be formed, there should also be certain requirements. The point is, what are the requirements? Proton gradient and an enzyme. ঠিক আছে সেইখিনিও ডিটেইলছ আমি ফটোসিনথেসিসত কম কৰ পৰা হয় কেনেকৈ হয় as of now protons বিলাকে generally they will help in the formation of ATP, right? And this one will generally help in the formation of the reducing cofactor, right? So ultimately just forget about all this, come back to this one. What we know that whenever an organism prepares its own food, then obviously they will require a source of electron and proton, right? If the source of electron and proton is an inorganic substance, that group is called as a lithotrope, right? Litho mane ki rocks, right? Rocks ki hoy inorganic, right? So lithotropes ki hoy those organisms that have the source of electrons and protons from inorganic substance. Those organisms that has the source of electrons and protons from organic substance like carbohydrates, fats, proteins, etc. Ebilak bhangi pelai jeli electrons, protons tahate use kore, then that group would be called as what? Organotrope. Exactly the same information in this case also. This is a group of organisms that will prepare its food in the presence of certain types of chemicals. But in spite of that, they will require a source of electrons and protons. What is the source? If the source is inorganic, then it will be chemolithotropes. If the source is organic, it will be chemoorganotropes. Is the concept clear? Words kita buji paiso ne kiba confusion ase? New student? Understood? What are you achieving in the class? Or are you feeling sleepy? Early morning class, AC ase, meme story gaye ase. Please pay attention, right? As it is, you have less time, right? Did you understand? Should I ask you a question or I shouldn't ask you a question? No, please sit down, please sit down, please sit down. क्या हुआ? क्लियर नहीं हुआ तो क्या कर रहे हो क्लास में? मैं क्या कर रही हूँ? नहीं कोई बात नहीं, डजंट मैटर. अभी तो ऐसा नहीं है कि तुम सब कुछ घर में पढ़ के आके तुम क्लास में आओगे, ऐसा तो है नहीं, है कि नहीं? सो यू नीड टू सर्टेन कंसेप्ट्स तो क्लास में भी तुमको वो करना पड़ेगा ना, यू हैव टू क्लियर इट ऑफ, राइट? सो जो पढ़ के आया, जो पढ़ के नहीं आया, जस्ट फॉरगेट. अभी जो मैं समझा रही हूँ वो समझ में आया कि नहीं, वो बात तो चाहिए ना, बिकॉज द नेक्स्ट टाइम यू हियर वर्ड कॉल इज chemolithotropes, then you need to understand, right? So please pay attention in the class, okay? Now you all are not kids. সৰু বাচ্চা তো নহয় যে মই উঠাই উঠাই তোমালোকক question কৰিম. আৰু if I start asking question one by one, you will also feel bad. Yes or no? No one feels good. কোনোবা question কৰিলে ভাল পায় নি? মোক যদি কোনোবাই question কৰে মই ভাল পাওঁ নেকি? I don't like, right? So no one likes if a question is asked, but if a question is asked, then the concepts might become clear also, right? So since you all are grown up students, please ask your own doubts, right? Let me not point out and say, please tell me the answer of this question, right? So nijor ji doubts ase, please ask 100 times, ase 100 times khuda. ঠিক আছে, কিন্তু concept clear kora. You don't have extra time to study at home now. You have very less time. Okay. So this part is clear, right? Next we'll go to the types of these, right? Photolithotropes are types ase, photoorganotropes are types ase, all will be divided into types. So what you do, either you can continue the chart or else you can put it into a separate heading. Heading ki likhiba? Photolithotropes boli heading likha. What is photolithotropes? I think the concept is clear now. Yeah, please ask. Kwa. Okay, explain koi dim mo point kita. Ha, tumi khali muk kuntu point buji poa nai okoman muk koi dile, it will be easier. Kwa. Etao buji napala niki? ওকে কোনকিটা পাইছিলা যোনখিনি পাইছা সেইখিনি ৰিপিট নকৰো হা সো কোনখিনিলৈকে পাইছা বুজি কোৱা কোনখিনিলৈকে পাইছা কেমোসিন্থেটিক আৰু ফটোসিন্থেটিক ওয়ার্ড দুটা বুজি পাইছা নে নাই পাইছা তাৰ পিছৰ ওয়ার্ড কিটা বুজি পোৱা নাই ফটোলিথোট্ৰফ কেমোলিথোট্ৰফ ফটো অৰ্গানোট্ৰফ কেমো অৰ্গানোট্ৰফ এইকিটা ওয়ার্ড বুজি পোৱা নাই ওকে শুনা হা সোৱা যিকোনো বস্তু এটাই যিকোনো অৰ্গানিজম এটাই যদি ফুড প্ৰিপেয়াৰ কৰিব লাগে ফুড প্ৰিপেয়াৰ কৰিব লাগিলে তাহাঁতৰ কিছুমান ৰিকুৱাৰমেন্টস থাকে আমাৰ যেনেকে ভাত খাব লাগিলে চাউলটো আনিব লাগিব চাউলটো ৰান্ধিব লাগিব তেতিয়াহে খাব পাৰিম হয় নহয় ঠিক তেনেকে প্লান্টস বিলাকৰো যদি ফুড প্ৰিপেয়াৰ কৰিব লাগে তাহাঁতৰ কিছুমান ৰিকুৱাৰমেন্টস থাকিব ৰিকুৱাৰমেন্টছৰ ভিতৰত ফুড প্ৰিপেয়াৰেশ্যন প্ৰসেছৰ ৰিকুৱাৰমেন্টৰ ভিতৰত দুটা ৰিকুৱাৰমেন্ট বা কেটামান ৰিকুৱাৰমেন্ট থাকে যেনেকে ইয়াতে আমি কি কৈছো যে যদি ফুড প্ৰিপেয়াৰ কৰিব লাগে তেতিয়া হলে ইলেক্ট্ৰন এণ্ড প্ৰটোনৰ এটা সোৰ্স লাগিব যেনেকে প্লান্টছত আমাক কাৰ্বন ডাই অক্সাইড এটমস্ফিয়াৰৰ পৰা প্লান্টস বিলাকে লয় তেতিয়াহে ফুড প্ৰিপেয়াৰ কৰিব পাৰে ৰাইট ঠিক তেনেকে ওয়াটাৰ লব তেতিয়াহে ফুড প্ৰিপেয়াৰ কৰিব লাগে ইট ইজ নট অনলি যে অকল কাৰ্বন ডাই অক্সাইড লব ওয়াটাৰ নলয় তেনেকে ফুড প্ৰিপেয়াৰ নহয় দুটা বস্তু লব লাগিব তেতিয়াহে ফুড প্ৰিপেয়াৰ হ'ব এইখিনিটো বুজি পাইছো ওকে কাৰ্বন ডাই অক্সাইড যে প্লান্টছে লয় কাৰ্বন ডাই অক্সাইড কি হৈ যায় CO2 ফৰ্মতটো সেলত নাথাকে এটমস্ফিয়াৰত কি ফৰ্মত থাকে CO2 ফৰ্মত থাকে প্লান্টছে কাৰ্বন ডাই অক্সাইডটো লৈ ললে সেলটোৰ ভিতৰত CO2 হৈ থাকিব নেকি নাথাকে যদি থাকিলে হয় সেল বিলাক যখন একদম পুরা গ্যাসিয়াস স্টেট গম বেলুনৰ নিচাকে ফুলি ফুলি থাকিলে হেতেন রাইট সো কাৰ্বন ডাই অক্সাইডটো কি হ'ব ফুডলে কনভার্ট হৈ যাব ঠিক আছে ফটোসিন্থেসিস হ'ব লাগিলে ওয়াটাৰটো লয় এটমস্ফিয়াৰৰ পৰা নহয় obviously by absorption through the roots water যেতিয়া লয় হে water molecule to যদি সেলৰ ভিতৰত ভৰি ভৰি গৈ থাকে তেতিয়া গুটেই সেলটো দেখোন swell up হৈ যাব water molecule টোৰ কিবা এটা হ'ব লাগে হয় নহয় so what happens to the water molecule it gets broken down right so if it gets broken down hey water molecule to pa ki produce kore electron produce kore protons produce kore aru oxygen produce kore right so ultimately ami ki kobo paru je jikono eta food preparation process ot amak hodai electron aru proton lagibo electron aru proton kor pora ahise tak ami ki boli koisu source যদি ইলেক্ট্ৰনছ আৰু প্ৰটোনছ কিবা অৰ্গানিক বস্তুৰ পৰা আহিছে অৰ্গানিক সাবস্টেন্স ভাঙি পেলাই ইলেক্ট্ৰন আৰু প্ৰটোন ফৰ্ম কৰিছে তেতিয়া তাক আমি কি কম অৰ্গানোট্ৰফছ যদি ইলেক্ট্ৰন এণ্ড প্ৰটোন কিবা ইনঅৰ্গানিক সাবস্টেন্স ভাঙি পেলাই প্ৰডিউস কৰিছে তাক আমি কি বুলি কম লিথোট্ৰফছ এই দুটা ওয়াৰ্ড ক্লিয়াৰ হৈছে ওকে সো ইন দিস কেস এই গুটে গ্ৰুপটোৱে ফুড কেনেকৈ প্ৰিপেয়াৰ কৰে ইন প্ৰেজেন্স অফ লাইট নে ইন প্ৰেজেন্স অফ কেমিকেলছ in presence of chemicals so eto hol the main line chemosynthetic dubhagot bhag korise on the basis of what on the basis of electrons and protons corpora paise jedi organic substance bhangi pela electrons and protons loise tetala tahatok ami ki boli kom chemo organotrophs word to ase so I think it's easy to identify right aru jedi eto group e electrons aru protons inorganic substances par loise tetal tak ami ki boli kom chemolithotrophs right so ultimately ki monot rakhiba just duta word jedi inorganic par loise lithotrophs jedi organic par loise organotrophs is it clear now okay কোৱা photosynthesis সোৱা জুনে photosynthetic আৰু chemosyntheticৰ ডিফাৰেন্স কি বা সিমিলাৰিটি কি সিমিলাৰিটিটো হ'ল যে দুইটা গ্ৰুপে কি কৰিব দুইটা গ্ৰুপে ফুড বনাব হয় নহয় photosynthetic গ্ৰুপটোৱে ফুড বনাব আৰু chemosynthetic গ্ৰুপটোৱে ফুড বনাব দুইটা হেতু সিমিলাৰিটি হ'ল নে নাই right? So moi aru ki koisu jedi food banai tetia hole source of electrons and protons lagibo hi photosynthetic e hok ba chemosynthetic e hok duitai to khwa bostu banabo no ar duitai ji hetu khwa bostu banabo duitake eta source lagibo electrons and protons or right? Photosynthetic bacteria bilake light lobo light or logot tahatok eta source of electron and proton lagibo chemosynthetic bacteria bilakok chemical reactions ba chemicals lobo aru eta source of electrons and proton leba hodai duta bostu lage right tumi ei bostu to kenike clear hoba photosynthesis or jedi eta reaction asile no the entire process could be depicted under one reaction thik kenike respiration buli koleo eta particular reaction asile photosynthesis or reaction to sabasun ki likha ase water plus carbon dioxide will give you the food hoine so duta requirement ase so photosynthetic bacteria bilakoru duta requirement ase eta hol light aru eta hol source of electron proton chemosynthetic bacteria bilakoru duta requirement ase eta hol chemicals aru eta hol source of electron and proton he karone duita group ore duta bostu thakibo clear welcome okay so etia now what we'll do is since the concepts are clear we'll be talking about the types of photolithotropes and photoorganotropes okay So if it is photosynthetic, then obviously it will undergo photosynthesis. If it undergoes photosynthesis, pigments should always be present, right? So if a plant is undergoing photosynthesis, tetia holeo tahator respective pigments bilak thakibo. I think you have heard something called as photosynthetic pigments, right? Whenever we talk about photosynthetic pigment, give me one example of a photosynthetic pigment. chlorophyll. Is it only chlorophyll that helps in photosynthesis or there are some other pigments also? Okol chlorophyll e thake ne aru beleg beleg pigments o thake jone niki help kore photosynthesis. Heard something called as xanthophylls, right? Carotenoids, right? So there are so many photosynthetic pigments, but the most important is obviously chlorophyll, right? So if you are talking about a plant or chlorophyll thakibo. If you are talking about bacteria undergoing photosynthesis, they will also have chlorophyll. But chlorophyll, I mean naam keneke koru. If you use the word only chlorophyll, this is something that is referring to the plants. If you are talking about bacterial chlorophyll, slightly different, then it is the chlorophyll that is present in case of bacteria. So bacteria also needs chlorophyll for the process of photosynthesis. So bacterial chlorophyll, obviously it will be present. Apart from bacterial chlorophyll, a specific type of a pigment, bacterioviridin and bacteriopurpurin, right? In case of purple, it's purpurin. In case of sulfur or green sulfur, green, it is bacterioviridin. So yarpra question keneke ahibo, identify the bacterial pigment. Right, bacterial pigments boli kole bacteriochlorophyll, bacterioviridin, bacteriopurpurin. These are the three words that you need to remember. Next, the most important question is about the example again, right? Give an example of a green sulfur photolithotropic bacteria, or give an example of a green sulfur bacteria. Give an example of a purple sulfur. Give an example of a purple non-sulfur, right? So examples again important. I think this part is over, right? So we'll go to that classification. Again here, if you are talking about green sulfur, green assay, so the pigment that will be present is bacteriochlorophyll and bacterioviridin. If you are talking about purple sulfur, bacteriochlorophyll and bacteriopurpurin, the pigments would be the same. Nitrifying bacteria by nitrification, heard about these words, right? Ki hoy, what is the thing that happens during this process? Nitrification boli koa loge lage ki hoy tate? Conversion of something into something, what into what? This is a process related with what? Nitrogen fixation in case of a green plant. Why nitrogen has to get fixed? Or what you understand by the word called as nitrogen fixation? Fixation mane ki? Ekdom simple ke unusable form of nitrogen is converted to usable form of nitrogen. Fixation mane? A conversion to kot hobo, soilot hoy no, so fix in the soil. In photosynthesis also something fixation takes place. What fixation? Carbon dioxide fixation takes place. Plants carbon dioxide loy. So cell to bhitor carbon dioxide humale, that is called as fixation, right? In the soil, the nitrogen or that got converted to the usable forms in the soil, that is called as fixation. So if you talk about nitrogen fixation, there are certain things that happen. What happens? Unusable forms of nitrogen gets converted to usable forms. What is the reason behind? Because plants cannot take in the atmospheric nitrogen directly. Why it cannot take in? Or who can take in? We know in the atmosphere there are abundant source of nitrogen, 78% nitrogen ase. So it is in a huge amount. Plants bilake directly, if they can take it, then it would have been so much of easier thing, no? Because nitrogen plants bilako kiho karne lage? Because nitrogen is a very important component, no? Sobote thakibo nitrogen. Ultimately, each and everything that need to form proteins, amino acids, everything needs nitrogen, right? So proteins fully kole, it is a building block of a cell. For that building block, you need to have nitrogen. Nitrogen is present in the atmosphere, but they cannot take it. A group of organisms who can take in nitrogen directly, kun hoy? Atmosphere or nitrogen to directly kune lobo pare? Microorganisms, right? Remember rhizobium? Rhizobium e ki kore? Nitrogen fixation not help kore. Means what? They can take in. Plants cannot take in directly. What is the reason? There should be something present in the microorganism that is absent in the plants. What is that something? A particular type of enzyme that is called as nitrogenase. Nitrogenase enzyme, if it is present, the organism can take the atmospheric nitrogen directly. Why? Again during that chapter. Because I mean mineral nutrition, what is the role of nitrogenase? How does it help in conversion, etc., etc. Right? Next is, in case of nitrogen fixation, we have talked something called as process like nitrification, ammonification, denitrification, nitrogen cycle asile no, tate ami ebu words poriso. Nitrifying bacteria helps in the conversion of unusable nitrogen to nitrogen, right? And if you are talking about unusable nitrogen, prothomote atmosphere jeto nitrogen thake, it gets converted to what? Okay, the first question, plants absorb nitrogen in which form? NO2 na NO3, nitrites na nitrates. Both puli kya ko bonwara? Because there are certain types of exceptional plants who will take in nitrites directly. It is always nitrate form that the plants absorb it from the soil. Atmosphere ki form ot asile? N2, right? N2 ki hole convert hobo lebo? NO3. So something should get added also, no? Oxygen add hobo lagibo, right? A process to kune help koribo, kisuman microorganisms will help, and those microorganisms, especially the bacteria, are called as nitrifying. What they will do? They will convert N2 into ammonia, ammonia into nitrites, nitrites into nitrates. Is the process clear? Atmosphere ki thakibo? N2. N2 will come and get mixed to the soil and form what? Ammonia. Ammonia will get converted to nitrites, nitrites will get converted to nitrates. This process, ammonia into nitrites, nitrites into nitrates, who helps? It's the aerobic bacteria, right? So details in that chapter, here just we are writing a summary. Right. So ammonia to nitrites by nitrosomonas, nitrites to nitrates, nitrobacter. This is the most frequently asked question. Question to kene ke kahe? The group of bacteria that helps in the conversion of ammonia to nitrites. You have to choose between the word nitrosomonas and nitrobacter, right? Nitrobacter always helps in the conversion of NO2 to NO3. Nitrites to nitrates, right? Can ammonia pra convert koribole kune help koribo? Nitrosomonas. What is nitrosomonas? What is nitrobacter? Nitrosomonas is a group of bacteria. Nitrobacter is also a group of bacteria. We are not talking about the individual bacteria. If I say Rhizobium, it's a name of an individual bacteria. If I say Bacillus, it's a name of an individual bacteria, but we are using the word called as Nitrosomonas. This is nothing but a group of bacteria, right? Similarly, Nitrobacter, we have certain examples of Nitrobacter, but that's not important. What is important? The name of the group, very, very important, right? Two groups of sulfur bacteria, right, depending on what is the function that they do. Okay, up till the portion that we have studied up today, one thing that we can say is plants undergo photosynthesis. That is something that is very common. There are certain bacteria that undergoes photosynthesis also. Not necessary each and every bacteria is photosynthetic, right? Bacteria buli kole bacteria is photosynthetic hobote enuka kotha nohoy. It is only a group of bacteria that is photosynthetic. So examples of photosynthetic bacteria we need to know. We have already discussed of green sulfur, purple sulfur, that those are examples of photosynthetic bacteria. Now plants undergo photosynthesis, certain types of bacteria also undergoes photosynthesis. What is the similarity between the two processes and what is the difference between the two processes? Obviously, there should be a difference between photosynthesis in plants and photosynthesis in bacteria. Similarity is, in both the cases, food would get prepared in the form of glucose or carbohydrate or starch, whatever you say. What is the difference? Pigments. Pigments are different, but the basic difference is something about their raw material, something about a gas that does not evolve, oxygen, oxygenic process and non-oxygenic or anoxygenic process. Oxygenic process boli ketia koba when oxygen is evolved, and that is obviously we are talking about plant photosynthesis, right? But in case of bacterial photosynthesis, oxygen does not get evolved, right? Plants photosynthesize korile oxygen evolve kore, bacteria photosynthesis korile oxygen evolve no kore. What could be the reason? One statement is clear: bacterial photosynthesis non-oxygenic means oxygen does not get evolved. Plant photosynthesis oxygenic means oxygen get evolved. Why? What is the reason behind? The hint is you have to remember the source of oxygen. Oxygen co-perform kore. Water perform kore. Water in the process of photosynthesis or as a raw material is used only by the plants, right? Plant or photosynthesis or homoiod, they will require carbon dioxide plus water will give you the food. This is a reaction for plant photosynthesis. Bacterial photosynthesis, since oxygen is not evolved, so obviously that source is not present. So what will be the reaction? Carbon dioxide plus something will give you the food, and what is that something? H2S. Right? Because they use H2S instead of water, oxygen does not get evolved. Right? Again, details in the photosynthesis chapter. So, the nutrition part is over, right. Autotrophic, heterotrophic, autotrophs divided into photosynthetic, chemosynthetic, photosynthetic, chemosynthetic again divided into lithotrophs, organotrophs, right. Types of organotrophs, types of lithotrophs, gutekitar examples bur dilu, right. Tar bitor maximum asked question is about this one. So, nitrifying bacteria bhalke poriba. Another question that is asked about iron bacteria also. As the name suggests, no, iron bacteria to obviously ki koribo, conversion on help koribo, ferrous F to F3, right. Next question kya hai, this is etu birat commonly asked, identify or give an example of a iron bacteria. I have written two examples. Obviously, first example of a question iman generally nahe, because whenever we hear the word ferro, we understand that it's obviously related with iron. That's a very simple question. Maximum question has been asked about the second example, right. So, identify a iron bacteria, etu ami poribo leibo, leptotrophs. Now, we will write certain points and I think it's almost over. So, in this portion again examples kini bhalke poriba. Heterotrophs also we have discussed the examples, right, autotrophs or heterotrophs or. out of the points that we have written, important key, aerobic nitrogen fixers, right? Can anyone tell me why is it important? Nitrogen fixers, no? Nitrogen fixation or help kore. The question is, nitrogen fixation is an aerobic process ne anaerobic process? Means nitrogen fixation occurs in presence of oxygen, in absence of oxygen. It is an anaerobic process. Whenever we talk about nitrogen fixation, it is an anaerobic process. We are going to talk about nitrogen fixation when we will be discussing blue-green algae also. And blue-green algae main kam ki fertility of the soil. By what? By nitrogen fixation. We will discuss that time. As of now, remember, nitrogen fixation is an anaerobic process. In presence of oxygen, nitrogen cannot get fixed, right? But we have certain types of aerobic nitrogen fixers also. This is all for today, right? In the next class, what we will discuss is, we will be talking about the last point about the life process, that is reproduction, right. So, I think we will have a class only after puja most probably. Ketia pra bondho dise na janu. Thik ase? Jedi hoy, then we will have a class on today is Friday, as a Saturday. So, biological classification would be on Wednesday, right. Morning classes we are having biological classification. So, the next morning class will be biological classification and we will be talking about reproduction. So, any doubts? Online students, kiba doubts asil niki? Clear, no? Okay then, thank you.